Chapter 5. In this chapter, we're going to be exploring the cell biology of both bacteria and eukaryotic cells. In our opening case study, we see a woman named Sharon who likes to spend time outside on her wooded deck in the Northeast, and she ends up getting an infection called Lyme disease. This is caused by a bacteria named Borrelia burgdorferi. You can see here in the scanning electron microscope image. Once this bacteria is delivered to the body through a vector called the deer tick, it can cause this bullseye-shaped rash. So you see the tick bite was in the center of this. You see sort of a darker red, and then there's these concentric circles on the outside. This is a pretty characteristic rash for Lyme disease, but it's not always present. It's only present about 30% of the time. This particular bacteria, once transmitted to the human through the tick, can cause crippling arthritis, it can cause meningitis, and can cause long-term chronic problems. So early identification and treatment with antibiotics is necessary. In this case, because Sharon presented with the rash, she was treated relatively quickly. Now, why are we opening the Chapter 5 case study with this particular bacteria? Borrelia burgdorferi, which is the name of the bacteria that causes Lyme disease, has a very characteristic corkscrew shape. So you see here it's spirochete shaped. We've seen a bacteria that has this shape in previous chapters. Treponema pallidum, the causative agent of syphilis, also has this corkscrew-like shape. And in a chapter that we talk about cell biology, it's important to point out one of these specialized structures. So this is part of the axial filament that creates the filaments that go around the bacteria. You can see them here. And they are attached to flagellar motors. So this is a specialized kind of flagella that is rigid, gives the bacteria this shape, but also allows it to move in a corkscrew-like fashion through the cell. So it's an example of one of those tightly controlled cellular structures in the bacteria that can have more than one function. Now, the higher order learning outcomes for Chapter 5 include identifying those key differences in prokaryotic and eukaryotic cells that we can utilize to combat infection. So human cells don't have these axial filaments and this type of flagella, and so therefore we could target this structure in bacteria and not harm humans. And that's kind of what we want to think about as we go through this chapter. Our second higher order learning outcome is to describe how bacteria have tightly regulated cellular structures that provide those unique functions to the bacterial cell. And again, our opening case study shows an example of this. Number 3, correlate the structure and the function of the cellular components in both prokaryotic and eukaryotic cells. So what are those cellular components made of? What is the chemistry of them? How are the structures arranged to help them do those jobs that they provide for the cell? Okay. So the common characteristics of bacterial cells is the first thing on our study guide, and we really need to appreciate that they have some unique features. They're listed over here on the left-hand side of the table, but before we start comparing, let's sort of highlight those common characteristics. So one, they have very complex cell envelopes, and your book defines the cell envelope as the membrane, the cell wall, and then any other structures outside of that cell membrane. So all of that together in a bacteria is very complex. The second thing they have is a small genome. So bacterial cells and prokaryotic cells have very small genomes, and that allows them to be very dynamic in how quickly they replicate and evolve. And then the third thing is they have those tightly coordinated cell parts. There's not a lot of room for redundancy, so bacteria will only have one part for every function. It's not going to have two different structures that do the same thing. So they're very, very efficient cells because they live as those single-celled organisms. This table here highlights the differences then between bacteria, which are prokaryotic, and eukaryotes. So bacteria are very small. We talked a little bit about that in chapters 3 and 4. The DNA is organized in a nucleoid, right, in bacteria, whereas it's in a tightly enclosed membrane space called the nucleus in eukaryotic cells. We've already mentioned the small genome. It's oftentimes a circular genome in bacteria, although they can have linear chromosomes. There's some examples of that. Whereas in eukaryotes, they're almost always linear chromosomes. They replicate and segregate during cell growth, and in eukaryotes, those processes are divided into mitosis and meiosis, so they have a little bit more complicated chromosomal replication. There are no intracellular membranes in bacterial cells. There's one example of thylakoids, but that's sort of a folding in of the same membrane, whereas eukaryotic cells have a lot of internal compartments or organelles. Here in bacteria, the cell wall, like we talked about already, is a complex part of the cell envelope, and the cell wall in bacteria is composed of peptidoglycan. Although some eukaryotes do have a cell wall, it never contains peptidoglycan. It's one of those great ways that we can target bacteria without harming human cells. And then when we talk about movement, we know that bacteria can move through rotary flagella, meaning they move sort of in a circle, and they could be driven by the proton motive force. Whereas eukaryotes, they may also have flagella for motility, but it moves more like a whipping action. Okay, so let's dive in a little bit closer to the structure of the bacterial cell. When we look at an EM picture of an E. coli cell, we can see a large collection of structures. Now remember that electron microscopy allows us to zoom in pretty tight, but even on that zoomed in image, there's really not a lot of differences here. We see sort of a lot of purple ribosomes, we see the nucleoid area here in orange, and we see parts of the cell envelope highlighted here in pink. There's not a lot of compartments within the bacterial cell. If we look at this cartoon or diagram description of the bacterial cell, we can see that as well. It doesn't have a lot of internal parts, but on the outside surface there's a lot of complexity. So look at all these different things that can be as part of the cell envelope, whereas the inside of the cell there's not a lot of different things happening. When we think about the chemical structure of all of these parts of the bacterial cell, we need to take a second and sort of appreciate that. So in chapters 4, we learned all about the chemistry of the macromolecules that we need to know. And so let's point out some examples here. So here we have the flagella, which is a structure that's anchored into the cell membrane, and this is primarily made out of protein. We know from previous discussions that the cell membrane, so this inner membrane, cell membrane here, is made out of phospholipids, those special molecules that are amphipathic and have the polar head group and the nonpolar fatty acid tails. We can see that in the case of a bacterial cell wall that has lipopolysaccharide, we can tell from the name here that it has part lipids and part polysaccharide or sugar, carbohydrate. So here's a lipid sugar component to the cell wall. We know that peptidoglycan can be found in the bacterial cell wall, so here we see parts of that in this blue area. And from its name, peptido, we can say that there's protein, and glycan, we can say there's carbohydrate. So peptidoglycan is a protein sugar component. Now, internal to the cell, we know that most of the cell's volume is made up of water, so water obviously plays a very big role here. We also have a collection of minerals and vitamins and ions that help regulate osmotic pressure and help provide the essential parts of enzymes. The enzymes that we can see in the cell, we see these RNA polymerase. We can see that highlighted over here. This is the turquoise in there. These green DNA binding proteins that help stabilize and manage the nucleoid area. Those are all made of protein. And we see this ribosome that's made up of both RNA and protein. So we see some structural RNA as well as protein here. And then these blue snaking chains are the DNA itself. So that's the nucleic acid component of the cell. On every one of these ribosomes, you'll see sort of, if you zoom in, you can see both the DNA and messenger RNA being made. So here we see RNA that's not acting in a structural fashion, but in an information fashion. So we'll talk more about how does DNA go from DNA to RNA to protein in another chapter, but pointing out the chemistry. Now, all of these white components in the cell then represent a lot of the enzymes and proteins that are needed by the cell. So again, those are mostly proteins that we can see in these white or cream-colored shapes within the cell. Those are going to be all the different enzymes that create all the different structures that the cell has. And you can see those polypeptides being made in this yellow squiggle here. That would be that primary structure of the protein before it gets folded into those unique structures. Okay, so that helps us understand the chemistry of how the bacterial cell wall is made up of. Let's zoom into some particular areas of the cell. So talking about the membrane in section 5.2, we need to remember that the membrane is made up of those phospholipid bilayers, so we see that here. But we know that not everything can just move through that phospholipid bilayer from the outside of the cell to the inside of the cell. So we do have to have those transport proteins, and they come in lots of different flavors, shapes, and sizes. Here's an example of one. And they help participate in the transport between the inside and the outside of the cell. That transport can be two different kinds. It can be passive transport, where molecules can either move through the phospholipid layer by itself, depending on their size and polarity, or they can move from areas of high concentration to low concentration through transport proteins. So passive transport does not require any extra energy or ATP by the cell, and it typically follows a diffusion gradient. Active transport, on the other hand, is used to actively transport things against a concentration gradient or transport things that require a much larger, more complex transport protein to move through. In that case, we need ATP energy. Right? This diagram shows us one example of how we create that ATP energy, and it's also an enzyme process that's linked into the membrane. Now, one other type of protein that's found in the membrane is a siderophore transport protein, and so let's take a second to talk about that. So bacteria require iron to do a lot of the cellular functions and create a lot of energy for the cell, and so we have molecules that travel outside of the cell, these siderophores, go out and bind iron from the extracellular environment and then transport it back into the cell. Now that iron transport process is really key to a bacterium's metabolism, and it's one of those unique places where we can target. The biggest source of iron in a human is our blood cells, and of course you can imagine that the organisms that can come out and steal our red blood cells' iron are much more pathogenic to us than other cells. So we block the action of siderophores with our immune system and with various drugs to help prevent bacteria from stealing and lysing our red blood cells. But this bacteria has a specialized transport process for moving that iron through the membrane. And then lastly, we have the proton motive force. This is a component of the cell membrane that's an enzyme component, and it relies on the hydrogen ion concentration. So when there's a high hydrogen concentration outside of the cell, right, we can drive this sort of, I call it a sort of turnstile. As the hydrogen bounces down the electron transport chain, we can add a phosphate back onto ADP to create ATP. And this becomes really important in cellular metabolism, which we'll learn about in future chapters. But this is how we generate ATP energy in the cell. It's the last stage of cellular respiration, and it relies on this important complex that's found in the membrane, which is why we're talking about it here. Okay, so let's start on the outside of the cell and talk about cell walls for a second. Here is an EM diagram, electron microscopy diagram, where you can see the bacterial cell wall sort of outlined here. And it's made up of two basic parts. It's got a peptide cross bridge, these little arrows, that are wrapping around and holding together these glycan chains. So when we think about peptides and glycans, we get the word peptidoglycan. Now, bacterial cells all have some peptidoglycan, and we'll learn about in a minute that some cells have more peptidoglycan than others. Peptidoglycan is never found in eukaryotic cells. So it becomes a very important molecule for us to study, understand, and to develop drugs to target. Here's a little more complex diagram, and you're not responsible for knowing all these chemical structures, but what you can see is that the glycan part of this is a repeating sugar. Right? So we have these two different sugars that are attached together, represented by G and M for acetylglucosamine and acetylmuramic acid. So these repeating carbohydrate sugars. And then here's that protein linkage, right? So here we see those polypeptides hooking them together. So we have the sugar and the phosphate, and it creates this sort of cage that surrounds the entire cell. It's important to remember that the cell wall is one continuous molecule, so it's this entire cage that serves as this protective barrier for the cell. Because bacterial cells live as single cells their entire life, they require this outside structure to help them regulate pressure and protect them from changes in their environment. I like to think about it as a cardboard box. So if the cell membrane is the latex balloon, and you shove a balloon inside the cardboard box provides a lot of structure and safety for that balloon, but it also gives it its shape. And so the cell wall is responsible for the cell's shape. It also protects the cell. Now, a balloon without the cardboard box, as you can imagine, is much more vulnerable to getting popped. Now, we know that the cell wall in bacteria can be arranged in a couple different ways. I've already mentioned that they both have peptidoglycan, so we see the peptidoglycan in this diagram here with the repeating sugars and the peptide links. Right, so we see that on this side over here. But this figure is really highlighting the differences between the gram-positive cell wall and the gram-negative cell wall. So the gram-positive cell wall has the cell membrane, and then right immediately outside of that is a thick layer of this peptidoglycan. So many, many layers of peptidoglycan. It has an S layer and some glycosyl chains. So a thick layer of peptidoglycan helps protect the gram-positive cells. In a gram-negative cell, we see a thin layer of peptidoglycan, and outside of that, we see this other outer membrane. So these lipids here look a lot like the lipids found in the inner cell membrane, but there are some important differences. Instead of just a glycosyl chain, they have a specialized lipopolysaccharide molecule here called LPS. This particular molecule turns out to be incredibly important to us during pathogenesis because we create an enormous inflammatory response to LPS, and so we call this an endotoxin. This molecule is particularly toxic to our bodies. And so gram-negative bacteria, not only do they have a different drug targets are going to work differently on the gram-negative bacteria, but the presence of this outer membrane and this LPS creates different outcomes medically for patients. So understanding the difference between these two cell walls is very important. Here in the EM picture, you can kind of see that. So we have the inner membrane, we have this peptidoglycan, and this outer membrane. In the gram-positives, we just see the inner membrane and a thick layer of that peptidoglycan. Now, one of the reason why we call them gram-positive and gram-negative is because the gram stain helps us determine which structures are present in the bacterial cell. The crystal violet is trapped by this thick layer of peptidoglycan, whereas in the gram-negative cells, because most of the structure comes from lipids, the dyes are allowed to escape because we dissolve away these lipids during the decolorizing step. So if we go back and think about how the gram stain works, we can tell how these structures allow those colors to occur. Okay, so we have a case study here. This is a 13-year-old boy who presented with a swollen nose and lips. Upon examination, he had some skin lesions that had loss of sensation, so they were sort of a numb area. When they did a skin scraping, they found bacteria that looked like this. Turned out that this particular bacteria is called Mycobacterium leprae, and it's the causative agent of leprosy. Leprosy is caused by this bacterium, Mycobacterium, that has a very unusual cell wall. And because of that, it only grows in areas that are at low temperature. So we'll see it on the nose, lips, and outer extremities, the tips of the fingers. So in this heat sensing diagram, you can see how the nose is much cooler than the rest of the body. And here's an image of a man that has some advanced stages of leprosy. It does, over time, start to erode away the tissues in the body, and so the nose starts to flatten. So leprae comes from Latin meaning lion's face, because the nose starts to shorten. This particular bacteria only infects about 5% of those that are exposed, and it can go many, many years with no symptoms. The treatment, once it's discovered, is 2 years of antibiotic treatment. So that's a very, very long time. Now, why do we have this bacteria here, and how does it relate to our discussion of the cell wall? Mycobacterium have a specialized type of cell wall, so they have some additional complexity and components in their cell wall. And that makes this bacteria incredibly resistant to getting eaten by our immune system, to disinfectants, and to antibiotics. So it has this extra protective layer. We can see that here in this diagram. So we see our cell membrane down here, and again we still have a layer of peptidoglycan, as all bacteria do, but then on top of that we have this whole collection of other types of lipids and mycolic acids and other molecules that help protect this particular bacteria. Now, all of that protection comes at a cost to the bacteria. This bacteria does not take in nutrients very well either. So the bacteria divides about once every 6 weeks. It grows incredibly slowly. So this bacteria is very difficult to treat, and it takes a really, really long time to cause signs and symptoms in the body. So it's important to mention that although most bacteria fall into the gram-negative and gram-positive categories, there are some diversity in the types of cell walls that we see. All right, so let's zoom in a little bit and talk about the DNA nucleoid area. So this diagram, this EM picture here, is showing a human cell that's being infected by E. coli cells. So we see these E. coli in yellow here, and we see this eukaryotic cell. Now, this is a great diagram to sort of highlight the differences in how we store the DNA in each type of cell. So in the yellow E. coli cells, the nucleoid area is basically this bright yellow stuff, right? So it sort of takes up most of the cell, and it's right there at the surface. In eukaryotic cells, the DNA is the purple here, and it's hidden away by this whole cytoplasmic area. So this is just a good reminder that both prokaryotes and eukaryotes deal with their DNA a little bit differently. Okay, so again we see this nucleoid area in purple, and we see the ribosomes there. And if we look at a diagram of what that looks like, we see this big circular genome of DNA, and it's packaged up and sort of wound around with these DNA binding proteins in green, and they form these sort of domains where they sort of loop back on themselves. And it's the bacterial cell's way of organizing and compartmentalizing the DNA. The whole DNA molecule is anchored to the cell membrane at the site of the origin of replication. Sometimes this is abbreviated the ORI. This is where replication begins, and it will divide the cell in two directions. So it'll start copying this way, and it will start copying that way to create two copies of the DNA during cell division. So the bacteria is organized in a way to help it copy its DNA very quickly and have cell division in an efficient manner. We just talked about Mycobacterium that takes 6 weeks to divide, but an E. coli cell can divide once every 20 minutes. So again, the DNA is organized for efficiency, not only in using the DNA for a source of information, but also in replicating. When we think about how the bacterial cell divides, we can see a picture of that here. So here's the origin of replication, the DNA copies, the bacteria divides and splits in half, and we have two daughter cells. So this is an incredibly simple process called binary fission. It's much simpler than the eukaryotic cell, which undergoes meiosis and then mitosis to divide its cell. No phases to memorize here in bacteria land. Okay, so we have another case study here where we're going to be learning about MRSA. So here a 23-year-old college student is getting ready to go out on an internship and notices after visiting the doctor that she has a large swelling on her leg. The swelling eventually erupts and a large abscess occurs, and she goes to the doctor. The doctor, after initial investigation, diagnoses her with MRSA, multiply resistant Staph aureus. And you can see here a picture of that lesion. It's pretty ugly and gross looking, typically filled with really purulent pus. And it can be very difficult to treat because this particular bacteria likes to collect antibiotic resistance genes. So treating the bacteria with antibiotics can be very problematic. MRSA can be found in a hospital setting, like was acquired in this case study, but it can also be found in the community as well. Now, why are we talking about MRSA in this particular part of our chapter? It's because we're thinking about how bacteria divide. And the way that the cell divides, or what we call the septation of the cells when they sort of split into two, tells us a little bit about how they're arranged. So if the bacteria divides in one single plane, it will just pinch off like little sausages and create this long chain of cells. So we'll see that in bacillus arrangement often, and we'll see that in streptococcus. So strepto means chain, sort of a long chain. Here in these EM pictures, you can appreciate that this particular staphylococcus arrangement isn't really ordered like a chain. It looks more like a cluster. And that's because the septation of these cells happens randomly and at lots of different times. So these two cells, as the cells septate or divide, they pinch apart and stay somewhat attached. And then the next plane of division can be somewhat random. So it might divide here this way, it might divide that way, and that creates that cluster-like shape or arrangement. And we use that as a diagnostic tool. So when they swab your throat for a strep, they're going to say, okay, here I see a cluster of cells, you don't have strep. Or I do see a long chain of cells and you do have strep. So that arrangement can be really important diagnostically. All right, let's talk about some specialized structures. So the purpose of some specialized structures in the bacteria, one of the jobs a bacteria has to do, we learned about in chapter 2, is to adhere to the surface of their host or to the surface that they're attached to. So bacteria have a couple of different ways to adhere. Most of them involve this pili or pilus structure. So pili could be one single long tube that attaches a bacteria to a surface and allows it to move in a rudimentary sort of grappling hook-like motion. Or there can be really short multiple projections of pili called fimbriae that allow it to adhere as well. Some bacteria can adhere using a stalk-like structure, which we don't have a picture of. And those are the ways the bacteria are going to be able to attach and hold on to the surface. So again, bacteria that can attach to a surface can be more pathogenic than those that don't have that structure. One other job for the pili, and you can see that here, is the bacteria can attach to each other. So during a process called conjugation, a bacteria can extend out a pilus and grab onto another cell and hold the cell tight while DNA is exchanged. The last specialized structure we're going to talk about for bacteria is the flagella. So here's a beautiful EM picture of a salmonella cell with these long flagella. Flagella are protein tubules that are used for movement. So they're not used for attachment, they're used to move in the cell. And we talked already about they have that rotary structure where they move in a circle. That structure is anchored into the membrane. So here's a diagram and here's an EM picture of that. So it's got a complex sort of anchored structure. And we know that even if a cell has multiple flagella, they can move them in concentrated fashion. So they can move counterclockwise towards an attractant. So we call that chemotaxis, moving, the taxis part, towards a chemical. Right? And they can do this sort of random tumble motion, and their sensors here, these molecules that sense the chemical that they're trying to find, notice when they get closer. So they do this run and tumble where they swim and then they kind of check out the environment and then they swim and they say, oh, we're not very close. They swim, getting closer. So sort of like a coordinated game of hot-cold. That's how bacteria can move towards a particular chemical or signal that they are interested in. Okay, so our last section here, we're going to touch briefly on eukaryotic cell structure. Now we deliberately don't go into this in detail. Most of the time a basic biology course will have comprehensive structures of the eukaryotic cell, so we spend most of our time in this chapter talking about bacteria, but it's important to remember what those eukaryotic cells look like. So here you notice that on the outside of the cell, there is not a lot of complexity. So there's no real complex cell envelope in eukaryotic cells. But the inside of the cell is more complex. And so we have various compartments, and we call these organelles. So we'll see the nucleus, which holds the DNA, the Golgi apparatus, which helps make a lot of the membranes and transport molecules, the mitochondria, the endoplasmic reticulum that comes in both rough and smooth. We'll see the lysosome, which is used for digestion, and peroxisome as well. And in this diagram, we don't see chloroplasts, but we mention those as well because a lot of eukaryotic cells contain those like algae. So when we think about the endomembrane system, this is the coordinated use of membranes within the cell. So think of it sort of like a packaging and delivery system. And most of that comes from this Golgi apparatus here. But we know that information and proteins get made in the information for that comes from the nucleus. The proteins get made here in the endoplasmic reticulum. Those components tend to get processed and packaged in the Golgi and then move out to the cytoplasm. So there's this sort of direction of flow of material from the nucleus all the way to the outside of the cell. And that's powered here by the mitochondria. The DNA, you can see, is stored in this nuclear membrane. So this is a special compartment that the DNA is kept in, and that there are holes in that, or pores, we call them nuclear pores, in that nucleus. Immediately outside of that is the endoplasmic reticulum and that Golgi apparatus. The nucleus takes up a large part of the eukaryotic cell because the chromosomes in the genome of a eukaryotic cell are much, much larger than that of a bacterial cell. Here we see another diagram of that double membrane, the inner nucleolus, and then the outside of the endoplasmic reticulum. Now, where did these organelles come from? The endosymbiosis is sort of a theory that we have about how these different organelles came to be inside of the eukaryotic cell. So the mitochondria and the chloroplasts that we think about in the eukaryotic cell came from the membrane of a smaller cell. They tend to be the same size and shape as a prokaryotic cell, and they have a lot of the same characteristics. So it's thought that a larger cell engulfed a smaller cell and sort of made it do the work for it while providing protection. So here we have the mitochondria. It has a double membrane, and interestingly, the inner membrane is much larger than the outer membrane, and all this membranous surface helps it do cellular respiration, which we'll talk about in later chapters. It provides a lot of surface area for those enzymes to line up and create energy for the cell. The chloroplast also has a lot of inner membranes, but these act like little solar panels that help collect the energy from light and turn it into sugar. And the eukaryotic cell also has flagella that's used to move. So this is a structure that's found in both prokaryotes and eukaryotes, although there are some distinctive differences in the eukaryotic flagella. Another way that eukaryotes can move is through the use of cilia. So these are short, hair-like projections that are used for movement. So in bacteria we have fimbriae that are used for attachment, but in eukaryotes we talk about cilia that are used for movement. And then our last specialized structure in eukaryotes is the pellicle, or not the last, second to last, is the pellicle. And this is a structure on the outside of the cell membrane that can be used for protection in those single-celled organisms that are eukaryotic. And then lastly, we have the contractile vacuoles. This is a way that cells can bring in water. And you can see here that it has this sort of star, sun-like shape where the water comes in and then it pushes the water out of the central vacuole here. This is an example of sort of a paramecium that's using that. Alright, so that is our tour of the cell, both prokaryotic and eukaryotic. So look at those study guides and be prepared to discuss further.